Hi, welcome to this next video lecture in your general biology course. This session is entitled Biomolecules Part 1. So we want to begin looking at the various types of molecules that make up living things. So let's take a look at our learning objectives for this session. First, we'll want to talk about hydrocarbons and functional groups. We'll see that most biomolecules are made up of the elements hydrogen and carbon, with several different types of atoms added to that to confer specific properties. We'll want to make a distinction between monomers and polymers. Monomers will be single molecules. Polymers will be chains of molecules linked up, a very common feature in living things. And then we'll want to see how these polymers are linked up, and for that matter, how they're disassembled after the fact. So we'll look at dehydration synthesis and hydrolysis of polymers. So in the previous videos, we've been talking about this question, what is life? And we came up with these 6 features that we said describe life. And for something to be a living thing, it needs to exhibit all 6. We also said this would be the backdrop for the entire course this semester as we begin to explain each of these in a bit more detail. So we want to start with the idea of complex organization. So if you recall this slide, I mentioned that living things exhibit many layers of complexity. This figure starts up there in the left corner with the biosphere and then breaks it down into simpler and simpler things. But what I want us to do is actually start at the very reduced level there, at the level of atoms and molecules, and we'll try to work our way up. So we'll start with atoms and see if we can build molecules, and then build more complicated molecules, and eventually start to build the parts of cells. So we're going to start at the bottom and work our way up. Recall we mentioned that new properties may emerge as we move to higher levels of organization. So we'll try to acknowledge some of those emergent features along the way. So again, we want to start at the very bottom and look at the very atoms and molecules that make up living things. So most biomolecules at some level consist of hydrogen and carbon, and we call that a hydrocarbon skeleton. I'm not referring to the skeleton of your body, the bones. I'm talking about the skeleton of a molecule itself. So most biomolecules consist of a carbon backbone with hydrogen atoms attached. So what do we mean by this? Well, here's an example. So this is a simple substance called propane. It's got a 3-carbon backbone that you can see there. There's 3 carbons linked up in a chain. And then we've put hydrogens in all the other locations. Now recall from your other chemistry backgrounds that carbon will generally form 4 covalent bonds. So if you notice, there are 4 sticks around each carbon representing those covalent bonds. And hydrogens only form 1 covalent bond. So each hydrogen just has 1 bond associated with it. And we would call it a hydrocarbon skeleton because there's nothing here but hydrogens and carbons. Propane, by the way, is often in a gaseous form. This is what you use with your gas grill out on your patio to cook with in the summertime. But that's a very simple carbon-based molecule. So we could add 1 more carbon and throw a couple of hydrogens on there to fill in the extra bonds, and we now have butane. Also a combustible substance. This is often found in cigarette lighters, butane lighters. So it's very similar to propane. We're just increasing the hydrocarbon skeleton by adding a carbon, still keeping it linear. But these hydrocarbon skeletons can also branch. So notice in this molecule, isobutane, we've let an extra carbon come off the middle there, but we're still using hydrogens to fill in all the missing bonds or the extra bonds that the carbon will need. So we can have straight chains, we can have branching, we can even have double bonds between carbons where they share 2 pairs of electrons. So notice the double bond here in this butene molecule. And since there's a double bond there, that eliminated the need for a couple of hydrogens because, again, carbons get 4 total bonds each. So we could have double bonds. You could even have the hydrocarbon skeleton in a circle or in a ring. So this substance called cyclohexane, the carbons are linked up into a circular formation there. And again, putting hydrogens to fill in all the extra bonds. And even ring structures can have double bonds. So here's a fairly common substance. Maybe you've heard of this called benzene. But notice the double bonds between carbons, which also meant we had to eliminate some hydrogen atoms to maintain that 4 covalent bond pattern with the carbons. Now, certainly you don't have to memorize these substances. I'm just showing you some examples here to let you see how carbons can get linked up to form these hydrocarbon skeletons. But one thing you should be able to recognize from these is that because every bond you see here is a carbon to a carbon or a carbon to a hydrogen, those are all non-polar covalent bonds. Those are all equal sharing of electrons. And as a result, every molecule in this figure is hydrophobic or non-polar. So even though the structure is changing a little bit and the double bonds and the branching in the circles, all of these molecules share in common the fact that they are hydrophobic and non-polar because there's nothing but carbon-carbon and carbon-hydrogen bonds. So although we can build some different shaped things, we're not getting a lot of variety of chemical properties. And this would not be complex enough to build anything remotely close to life. Life is so complex that it needs the ability to perform a lot of chemical tasks, and that requires chemicals with a lot of different properties, not just hydrophobic molecules. So how can we take these hydrocarbon skeletons and make them have additional chemical properties? Well, what we do is we add what are called functional groups. So functional groups are clusters of atoms attached to carbon skeletons that confer a particular chemical property. So it's a little group of atoms that come together as a unit, and you pop it on to the hydrocarbon skeleton, and this little cluster, this little unit, brings with it a new chemical feature. So let's talk about some examples of functional groups and the ones that are most important in living things. The first one we'll mention is called the hydroxyl group. It's pretty simple. As the name implies, there's a hydrogen and oxygen involved here. So you've got some sort of a hydrocarbon skeleton, and then somewhere on there you add an OH, the hydroxyl group. Here's another representation of it with a ball and stick model. And this is a functional group that we find very commonly in carbohydrates. We're going to talk about carbohydrates more specifically later. For now, we can allude to the fact that it's things like sugars and starches and cellulose and things like this. In fact, here is a carbohydrate molecule. It's called glucose. Molecular formula is C6H12O6. And we're going to learn a lot about glucose later. You'll know this structure pretty well. But what I want you to notice at the moment is that it's sort of a ring of carbons a bit, and we've put hydrogens in many locations, but notice how many places where we've put the OH group, the hydroxyl group. So that's going to bring some new chemical properties to this molecule. In fact, it will no longer be hydrophobic. That carbon to oxygen bond that bonds the OH onto those carbons is a polar bond. So in fact, glucose is extremely polar, extremely hydrophilic, loves water, whereas a basic hydrocarbon ring would be repelled by water. So the hydroxyl group's the first example we've got. Another one is called the carbonyl group. This is where you've got a carbon double bonded to an oxygen somewhere in the molecule. And we will see later that the carbonyl group shows up in lipids. Another one that's going to be very important is the carboxyl group. You can see what it looks like here. We've got a carbon double bonded to an oxygen, so at first it looks a little bit like a carbonyl, but in addition to that double bonded oxygen, it has a hydroxyl. So it's almost like you're putting the first two functional groups together to create a new functional group called the carboxyl group. This is one that we're going to see very common in proteins, and although I'm not writing it up here on the slide, it's important to know that the carboxyl group makes a substance an acid. So we'll see examples of this later, but it might be important to note that carboxyl groups makes things acids. Another group is the amino group. It sounds a little bit like ammonia. Ammonia would be NH3. This is NH2. So notice there is a nitrogen with two hydrogens attached to it. Nitrogens always have 3 bonds around them. And I probably should have mentioned with those previous that oxygens will usually have 2 bonds around them. But nitrogens have 3, so this is the amino group, and we will also see it in proteins. Maybe you've heard of something called an amino acid before. Well, amino acids have both the amino group and the carboxyl group on them. And again, we'll learn about those here shortly. And then the last functional group that I want to mention is the phosphate group. This is where you have a phosphorus with oxygens going around it. And I mentioned just a second ago that oxygens will need 2 bonds, but notice 2 of the oxygens only have 1 bond, and that's why you see that negative charge next to them. The top oxygen and the one on the far right there, they've each got a negative charge because they're lacking 2 complete bonds. And that's what makes the phosphate group so important. It's very negatively charged. Those 2 oxygens have true ionic negative charges. So if you put a phosphate group onto a substance, you will have made that substance negatively charged, and that will also make it very hydrophilic, very polar. So we'll see the phosphate group show up quite a bit this semester, altering the features of molecules. So again, we start with a hydrocarbon skeleton, which is fairly simple and basic and non-polar, and then we can start popping on these functional groups to create very unique, complex molecules, similar to the glucose that you see here, but there's many other examples that we'll get into. Now, once we've got a hydrocarbon skeleton and some functional groups attached to it, we may have generated for ourselves what's called a subunit molecule, and then we can link the subunits together to form what are called polymers. It turns out that most of the biomolecules in living things are polymers, chains of subunit molecules. So for example, here is a chain down here of, how many did I use there? 5. So we got 5 subunit molecules all linked up to form a larger molecule that we're calling a polymer. So what type of subunits are we linking up? Well, there are 4 main types of polymers in living things. The 4 main types of polymers are carbohydrates, proteins, lipids, and nucleic acids. So all living things, whether we're talking about fungi or plants or animals or bacteria, are made up of carbohydrates, proteins, lipids, and nucleic acids. But these polymers themselves are chains of subunit molecules. So in the table, it is showing you what it is we need to link up to form these various types of polymers. Carbohydrates, for example, are chains of saccharides. Saccharides is just a sciencey word that means sugars, and I've already shown you a picture of a sugar. We looked at the molecule glucose. So there is one particular type of carbohydrates, like starch. Starch is a polymer of glucose. So what if we just link glucose to glucose to glucose to glucose to glucose? Now we have a polymer. We've linked up all the monomers or subunits. So subunits are also called monomers, single units. But I linked them all up to form a polymer. And in this case, I'm using the exact same subunit each time. Glucose, 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 glucose. But there are different types of saccharides. And we could link different types of saccharides together to create very unique types of carbohydrates. We'll talk about some of those types of saccharides a little bit later. The next type of polymer are proteins, and in proteins what we're going to be linking up are amino acids. We'll learn that there's about 20 different types of amino acids, so we can link them up in all kinds of orders and combinations to create all the various types of proteins. You've probably heard of some different types of proteins. Hemoglobin, maybe collagen, keratin. These are some common proteins, and they're made by linking amino acids up to form the chains, the polymer chains. I should also mention that in this figure, just for the sake of space, I created a polymer that had 5 subunits. Proteins may have several hundred amino acids linked together, so these things can get very complex. The third type of polymer are lipids, and we will see that we're going to link fatty acids together to form lipids. And then finally, the fourth category of polymers are nucleic acids, things like DNA. And DNA is made by linking up subunit molecules called nucleotides. So the main thing I'm hinting at here is that most of the molecules that make us up, these are polymers, carbohydrates, proteins, lipids, and nucleic acids. And polymers are chains of subunit molecules, and there's a lot of different types of subunits, and that's how we can create such a wide variety of polymers themselves. But how do we get these polymers linked up and assembled in the first place? Well, each monomer, and again, monomer is what we mean by subunit, each monomer, whether it is a saccharide or an amino acid or a fatty acid or a nucleotide, we can draw it very generically like this. Assume that the purple sphere represents all the hydrocarbons and the functional groups and all that kind of stuff. That's like the rest of the molecule of the saccharide or of the amino acid. But somewhere on that subunit, on that monomer, on that glucose, for example, we will find a hydroxyl group, an OH, and somewhere there will also be a hydrogen. And I want you to think about that for a second. If I have two H's and an O, I essentially have water. So what I'm really suggesting is that every one of these subunits, again, whether it's glucose, which is a saccharide, or an amino acid or fatty acid or whatever, somewhere on the molecule there will be an OH and an H, which also represents essentially like a water molecule. So there's one subunit. How do we link them together to form polymers? Well, there's a process that's used whether we're building carbohydrates or proteins or lipids or nucleic acids. The process that's used is called dehydration synthesis. This is the process by which polymers are assembled from subunit molecules. This is how, for example, we would link glucoses together to form that polymer of glucose. So let's say we already have a short polymer. Here's one that's got 3 subunits in it. And notice that it has the OH-H orientation on it. And we want to add another subunit to it, another monomer to this growing polymer. So here comes our unlinked monomer that we want to attach, and just as I said a second ago, it has an OH and an H on it. We will now use dehydration synthesis to attach that. So dehydration, that means losing water. When you dehydrate something, you're eliminating the water. So what we're going to do is take the OH and the H that are highlighted there, we're going to remove those. That's the dehydration part. And when that water is removed, the subunit at the end of the short polymer is going to link itself to the single subunit, and we now have a slightly longer polymer. We've added one more subunit to the chain. But notice that the longer polymer will still retain that OH-H orientation, so we could repeat this exact same process as many times as we need to and just keep adding more and more subunits to the chain. And again, this is how we would link subunits together, whether they're saccharides or amino acids or fatty acids or whatever. We do it by dehydration synthesis. So this basic process applies to all the various categories of polymers. And then the cell can sort of dictate which subunits are being linked together and what combinations and what order to create all the different types of proteins and all the different types of carbohydrates and so forth. So that's how the polymers are assembled. What about disassembly? How would you take this apart? Well, polymers are disassembled by hydrolysis. This is the process by which polymers are disassembled back into subunit molecules. If you think about the word hydrolysis, it literally means water breaking, using water as a pair of scissors. And it's essentially the exact opposite of dehydration synthesis. So let's say that we started with a longer polymer, and again, long is a relative term. These figures show you one with 4 subunits. A long polymer may have a couple hundred subunits in it. But anyway, we've got a polymer, and we now want to disassemble it by hydrolysis. So what we're going to do is use a water molecule as scissors. So water is going to come in and reinsert itself between the last subunit and the rest of the polymer, and that's what's going to knock that subunit off. So again, it's the exact reverse of dehydration synthesis. And if we want to keep disassembling that polymer that has 3 now, we would bring in another water molecule and knock the 3rd subunit off. And then we could bring in another water molecule and knock the 2nd subunit off. So we could disassemble this all the way down to nothing but individual subunits simply by using a water molecule each time to knock them off. So those are the processes. Whether you're taking apart a protein or a carbohydrate, we would use hydrolysis. Sometimes you will hear hydrolysis referred to as digestion. So if we want to digest a polymer, if we want to disassemble it into these subunits, we would use the process of hydrolysis to do that. Now, when I think of dehydration synthesis and hydrolysis, I cannot help but think of this pet ball python that I've got. So this is the ball python, very young. Well, literally, it's a baby. You can see how small it is. It literally fits in your hand right here. But after a few years, suddenly there was a whole lot more snake. And the question is, where did all of this snake come from? Well, I know the snake is made up of proteins, carbohydrates, lipids, and nucleic acids. After all, protein is what makes up snake scales and snake muscle and so forth. So I know it's made of those molecules, but where did it obtain all of those new snake proteins, new snake carbohydrates, and so forth? Because the only thing that's gone into its mouth are rats. That's what this snake eats. Turns out, though, that the rat is also made up of proteins, carbohydrates, lipids, nucleic acids. They're just slightly different. It's the mouse or the rat version of hair, the rat version of muscle tissue, the rat version of carbohydrates and DNA and so forth. But that's what goes in. So in goes the rat, and then what the snake is going to do is swallow that thing down into its digestive tract. So now we've got this rat down in there, and the snake's digestive system will now use hydrolysis to disassemble all of the various polymers making up the rat. So by hydrolysis, the rat will become disassembled into individual amino acids, individual saccharides, individual nucleotides, and so forth. And then, using dehydration synthesis, the snake will take those subunit molecules and, using dehydration synthesis, reassemble it as snake molecules. So you can take a rat apart and hydrolyze it down into monomers and then reassemble those monomers in new combinations, and now you've got snake muscle, snake skin, snake tongue, snake sugar, snake blood, all that kind of stuff. So that's one of the main reasons that living things eat, is so they can digest or hydrolyze what they ate and then reassemble it as their own versions of carbohydrates and lipids and so forth. In a way, it's almost like you've got a Lego house and you take it apart into just generic Lego pieces and then reassemble them into a Lego car or a Lego boat. Once you get the Legos disassembled into the generic subunits, you can then reassemble them in new combinations. And the same is true for you as it is for the snake. So here is a nice family here putting things into their mouths that they will then hydrolyze and disassemble. All the food you see on the table there is made of proteins, carbohydrates, lipids, nucleic acids. After all the food you eat is or was a living thing. So whether you're talking about the beans, the rice, the meat, the salad, the French fries, the corn, everything there at some point was a living thing. So made of proteins, carbohydrates, lipids, nucleic acids, and so forth. So we can eat those things, disassemble them, and then reassemble them as human stuff. So you could actually put this type of stuff in your body and disassemble it by hydrolysis, reassemble it by dehydration synthesis, and you can turn that Big Mac into human hair and human muscle. The problem is these food items are not exactly the right proportions of proteins, carbohydrates, fats, and so forth that our body is made of. So you may be ingesting extra molecules of fats and lipids, for example, that your body does not have need for. And so that's why this is probably not the best food to make your body out of, because you're going to have a lot of molecules that are not going to be necessary and are just going to be stored as fat, for example. It's best to make your body out of things that most closely resemble at least the proportions of nutrients that your body is. I would suggest building your body out of things like this. So you can have an unhealthy diet or a healthy diet, but one way or another, you're going to disassemble what you ingest and then reassemble it as your own body. So living things are made up of biomolecules. Those biomolecules are hydrocarbon skeletons with functional groups attached. Once you've created a simple biomolecule like a glucose, you can then begin to link those into polymers, and that's what starts to form the overall living thing. All right, well, that brings us to the end of this video session. In our next one, we're going to look at those classes of polymers in more detail. We'll look more specifically at carbohydrates and proteins and lipids and nucleic acids and see some examples and see what roles they serve in living things. But we'll get into that in the next video.